Okay, so welcome back to this next video in which we are discussing the 5-HT3 receptor and depression. Okay, so we're just discussing the structure of the 5-HT3 receptor at the moment. Okay, so I told you in the previous video that there are two main types of 5-HT3 receptor which are important in human physiology. Okay, now one of them is this 5-HT3A homopentamer, where the subunits that you have in all 5 of the slots are the 5-HT3A receptor subunit. Right, now that's a nice simple one because it's got the same protein in each of the 5 slots. Now the next one is more complicated and is less well understood. The other one that is found is what's known as the let me just pull this out a little bit so that I'm not bumping into the camera, 5-HT3AB heteropentamer. Okay, now hetero means different, so let me just finish squashing this in. So hetero means different, so it means basically that these subunits that you are making up your 5-HT3 receptor with are going to be different. So you're going to have some of one form and then some of the other form. Okay, so here is this same cartwheel drawing. So we need 5 subunits in the 5-HT3 receptor, like so. Now, this is called the 5-HT3AB heteropentamer, so that gives you a clue as to which of the subunits that are available to us, of which of these 5 subunits you're going to put in here. You're going to have a mix of the 5-HT3A subunits here, okay, and also the 5-HT3B subunits here. So you're going to have a mixture basically. You're going to take some of this one and some of this one and put them together to make this pentamer. Now, I would love to be able to draw a picture where I tell you exactly which of these are the 3A form and which are the 3B form. However, I can't. That hasn't been characterized. We don't even know what the stoichiometry is. We don't know whether you take 1 5-HT3A and 4 5-HT3Bs or 4 5-HT3As and 1 5-HT3B. We just don't know these things. All we know is that there exists one 5-HT3 receptor where you have 5 5-HT3A subunits all coming together to make a homopentamer, and then there is another one where you have a mixture of 5-HT3A and 5-HT3B subunits in it. That's all we know. So I'm afraid I can't draw this picture anymore for you, except for telling you that some of these will be 5-HT3As and some of them will be 5-HT3Bs. Right, now you will notice that these 2 receptors that I have told you, which are the ones that are known to have function within the human brain, they don't use at all the 5-HT3C or the 5-HT3D or even the 5-HT3E gene. So what on earth is the point of these 3? Well, basically, we think it's starting to emerge that there also exists heteropentamers where you have 5-HT3A subunits with one of these 3 here. So you'll have the 5-HT3AC heteropentamer. So let me write this down. 5-HT3AC. You might have this heteropentamer where you'll have some 5-HT3A subunits and some 5-HT3C subunits. Then there may also be the 5-HT3AD heteropentamer and the 5-HT3AE heteropentamer. So it seems that you always have to have some 5-HT3A subunits in your receptor, and then you can either make a homopentamer where it's all 5-HT3A, or you can have another one of these alternative 4 mixed in with it, but only one, so you don't start mixing more than 2 types of these receptors. So the rules basically are, when creating a 5-HT3 receptor, it seems to be you use always the 5-HT3A receptor subunits, and then you can either just use 5-HT3A subunits, or you can pick one other of these four here to mix in, basically, but only one other. Okay, but these heteropentamers down here are not fully characterized, they're not understood at all. I think it now is generally agreed that they exist, however. Right, okay, so that's the 5-HT3 receptor structure. So we have the two types now, these homopentamers and these heteropentamers. So when people are discussing 5-HT3 receptors, they generally just refer to the homopentamer, and they assume you understand that that is a homopentamer of 5-HT3A receptor subunits, and then they also refer to the heteropentamer, and it's assumed that you understand that that's a mixture of 5-HT3A receptor subunits and the 5-HT3B receptor subunits. Right. Okay, so now let's talk about opening of these 5-HT3 receptors. So for this, we'll go back to basics, because unfortunately it hasn't even been characterized where the binding site for 5-HT is on these receptors. So all we know is that 5-HT binds to these receptors, and that causes some sort of conformational change that opens them. So let's summarize what we know. So basically, I draw this here. So, we start off with a closed but resting receptor, and this is important because there are two closed states, not just one. There's probably more than two, but for our simple model we'll just have two. Okay, so we start off with what's known as the closed, but then you put a forward slash, and then you have resting state. Okay, so this is the state where no ions can move through the 5-HT3 receptor. However, if the ligand binds to the 5-HT3 receptor at the moment, it will cause that 5-HT3 receptor to go into the open state. Okay, so here are the here is our 5-HT3 receptor. I'm just drawing it very simply as a box with a pore through the middle. So at the moment it's in the closed/resting state. However, if a ligand binds, so this is the process of ligand binding, okay, then what will happen is the 5-HT3 receptor will open. Okay, so now it's gone into the open state where it's got a pore down the middle here. Okay, so now it's in the open state. Okay, so the ligand needs to be bound, so I'll draw some ligand bound here. So this little box here, remember, represents the ligand, and we don't even know how many ligand molecules bind to the 5-HT3 receptors, so we really don't know that much really about these receptors. We know they exist. Okay, so here it is bound to the receptor, and that causes it to open. Now, we do, however, know what they conduct when they open. They are permeable to sodium and potassium ions. Monovalent cations is what they like to conduct. So they are a cationic, selective, ligand-gated ion channel. Okay, now, the question is, what's the effect of opening a channel which is permeable to both sodium and potassium on a cell? Okay, well, to decide upon this, we need to consider what the concentrations of sodium and potassium are on either side of the membrane. So, usually, on the outside of the membrane, you have a sodium concentration that is around 145 millimolar. The intracellular concentration of sodium is usually around 12 millimolar. So you have a 12-fold gradient favouring the movement of sodium in. Then, the concentrations of potassium, the intracellular concentration of potassium is 155 millimolar, okay? And then the extracellular concentration of potassium is 4 millimolar, or thereabouts. These are rough figures, and if you look at different places, you'll find that these concentrations vary between different sources, but they'll all generally be around these numbers that I've given you. So these are the numbers I always use. Right. Okay, so you can see that potassium is much higher within the cell and low outside the cell. There's around a 40-fold, or nearly there, 39-fold gradient of potassium favouring the movement of potassium out. So the concentration gradient is favouring the movement of potassium out and favouring the movement of sodium in. And the concentration gradient for potassium is far bigger than the concentration gradient for sodium. So you'd expect the movement of potassium out to beat the movement of sodium in. That's why I've drawn the blue arrow bigger than the red arrow. So basically, you'd expect that if you open this channel, you'd get a net movement of positive charge out of the cell. However, that's not what is going to happen. And because we've forgotten to consider something. We've forgotten to consider the electrical potential difference across the cell membrane. Okay, so I'm just going to have a little discussion of what it means, what the electrical potential difference across the cell membrane actually means. So, basically, if you had a little man standing in the extracellular compartment, he can measure the electrical potential of the extracellular compartment. So basically, there is something known as the electrical potential scalar field in physics, which basically is a mathematical construction to understand reality. Okay, so you ascribe to every point in space a real number, which can be a positive or negative real number, and this is called the electrical potential. Okay, so every point in space has an electrical potential. It has this number ascribed to it. Right, so if you're in the extracellular space, you can measure the electrical potential there. So you can measure electrical potential, so I'll put a big E for electrical potential, and then I'll put a little e, like so, to denote that it's the extracellular electrical potential. He can then move into the intracellular compartment and measure the electrical potential intracellularly. Okay, and these two numbers will not be the same, basically. There will be a difference between them, an electrical potential difference. So, what you can then ask, what this little man can ask, is if he was to go on a journey, if he was to move across the cell membrane into the intracellular compartment, how much would the number on his machine that's measuring electrical potential, how much would that number change? And that is what is meant by the electrical potential difference from extracellular to intracellular, also called the voltage from extracellular to intracellular. Now really, people get sloppy. They should tell you the voltage from one place to another place. They should say the electrical potential difference from extracellular to intracellular. However, everyone assumes that you know that we're talking about from extracellular to intracellular. So people often drop this from extracellular to intracellular, and they just say the voltage across the membrane, or the electrical potential difference across the membrane, or the resting membrane potential, or something to that effect. But what they mean is, if a little man was to move from the extracellular to the intracellular compartment, how much would the number on his machine that's measuring electrical potential, how much would that change? So, basically it's going to give us the new electrical potential, which is the electrical potential intracellularly, minus the old electrical potential, which is the answer to this question: how much would the number on the machine change if I moved from extracellular to intracellular? Okay, so that's what's meant by the electrical potential difference across the cell membrane. Now, this is usually around negative 65 millivolts. Across most cells, the resting electrical potential difference across the cell membrane is usually around negative 65 millivolts. Now, what does that mean? That means that if you were to move from the extracellular compartment to the intracellular compartment, your electrical potential would go down. And it will go down by 65 millivolts. So the number on his machine will go down by 65 millivolts. Okay, so basically the intracellular compartment's electrical potential is 65 millivolts lower than the extracellular compartment's electrical potential. Now, both sodium and potassium cations are positively charged species. Positively charged ions want to go to the compartment with the lower electrical potential. They like being in places where the electrical potential is lower, which in this case is the intracellular compartment. It's lower by a whole 65 millivolts. So, the electrical gradient across the cell membrane is going to favour the movement of sodium. Well, it's going to favour the movement of cations in, full stop. Now, so it's going to help the concentration gradient of sodium, because the concentration gradient of sodium is pushing sodium in, but the electrical gradient is going to be in the same direction. So you're going to add a great big oomph onto the movement of sodium into the cell. So the movement of sodium is going to go up because of this electrical potential difference, whereas it's not going to help the movement of potassium at all out of the cell. Potassium wants to go out of the cell because of its concentration gradient, but the electrical gradient says no, come back into the cell. So basically, the concentration gradient points one way, the electrical potential gradient points the other way. It's kind of like in Game of Thrones. Whose word is taken, the queen's or the hand of the king's? But basically, who wins? Does the concentration gradient win, or does the electrical gradient win when they both want opposite things? Well, in this case, it's the concentration gradient. The concentration gradient is too strong. The queen outwits the hand of the king. So this is Ned Stark, this is Cersei. She wins. Basically, the concentration gradient is going to win, but it's going to be weakened by the electrical gradient. So the net movement of potassium out of the cell is going to be much smaller. So you're going to have to subtract this arrow off from this arrow to get a tiny little arrow. So you basically get a feeble little movement of potassium out of the cell. You just get a movement of potassium out of the cell because the concentration gradient does actually beat the electrical gradient. You'd have to take the electrical gradient down to around negative 85 millivolts in order for them to be equally matched, i.e. to get no movement of potassium out of the cell, for the electrical gradient strength to be exactly the same as the concentration gradient strength. However, at negative 65 millivolts, you're going to greatly weaken the concentration gradient. Okay, so you're going to get a massive great movement of sodium in, and you're going to get a tiny little movement of potassium out. So that means that the net movement of positive charge across the cell membrane when you open this cation selective ion channel is going to be into the cell. Now, if you move positively charged ions into the cell and out from the extracellular compartment, then that's going to raise the electrical potential of the intracellular compartment because you're bringing positive charge in. So this number here is going to go up. Okay, that's going up, and it's also going to reduce the electrical potential of the extracellular compartment because you're taking positive charge out of there. The positive charge doesn't just arrive in here from nowhere, it comes from somewhere, so you're pulling positive charge out of the extracellular compartment, and the positive charge was raising the electrical potential of the extracellular compartment, so when you remove it, that reduces the electrical potential of the extracellular compartment. So, originally, the intracellular electrical potential was 65 millivolts lower than the extracellular electrical potential. But if we're taking this number up and we're taking this number down, then the amount by which this one is smaller than this one is going to get less. So this number is going to become less negative. And when the electrical potential difference across the cell becomes less negative, we call that a depolarization. Okay, so opening these 5-HT3 receptors is going to result in a depolarization of the electrical potential difference across the cell membrane. Right, okay, and we'll continue this discussion in the next video.